Hello and welcome back for one more video in Chapter 8 of Biotechnology. So in this video we are going to talk about something a little bit different from the protein production and transformation process that we've been talking about in earlier videos in this chapter. We're going to talk about something that you wouldn't necessarily think to do, but is actually a very useful and very good thing to do, and that is after you have transformed cells with a plasmid, taking the plasmid back, essentially. So you may be wondering, what the heck am I talking about? Well, that should become clear here pretty shortly. So the idea here, as I said, is that once you have transformed cells with a particular plasmid like pGlow, it often is going to be necessary to extract that plasmid back out of the cell. But as I said, the question is why? Why would you want to do that? The cells are already transformed. Why do you want to get the plasmid back? Okay, so once you have completed a subcloning process, such as ligating a gene into a plasmid vector, once you actually have that plasmid and you're ready to transform cells with it, if you can get those cells transformed with it, you should never, ever, ever need to actually make that vector again. You shouldn't ever need to make the vector with your gene in it, because basically, if you can transform cells, you've got little plasmid creating factories. They will make their own plasmid once you've transformed with it, and basically we can let the bacteria, or other cells if you prefer, to make their own copies for us. So basically, once you transform the cells, you'll never, ever, ever have to make your own plasmid or buy plasmid again. You've basically got a lifetime supply, provided that you continue to transform cells and then retrieve the plasmid from the transformed cells. So again, the idea here is that we are tricking the transformed cells to not only make the protein product that is on the plasmid in that gene that we inserted into the vector, we're tricking those transformed cells into making many copies of the plasmid. And then once that's done, we can take those cells, grow them up, and then extract plasmid so that we can always have a healthy stock of that particular plasmid on hand. And from there, we can continue transforming new cells, getting more plasmid, getting more protein, transforming new cells, getting more plasmid, getting more protein, and we can really keep the gravy train rolling at this point. We shouldn't ever, once we have a plasmid, you should not ever have to get that plasmid or make that plasmid again. You can just get the cells to make more of it for you. So we mentioned something about this earlier, but many biotech companies like Zymogen or Qiagen actually sell kits for purifying plasmids. And these kits are a little bit different depending on what organism you're trying to get it from. But the name of what method you're using is usually going to depend on the amount and purity of plasmid that you desire. So the methods are all pretty similar, so just to keep things nice and simple, we're going to describe what's called a MiniPrep here, something that we will be doing in lab towards the end of the semester. So the process of doing a MiniPrep, if you just pay attention to this particular figure here, it looks a little bit involved, but it's actually very simple and should not take more than 30 minutes usually. So a broth culture of transformed bacteria are going to be necessary here, so you're going to want to start maybe a 1 to 3 milliliter culture of transformed cells, making sure, of course, that the broth contains the antibiotic to keep the selected pressure on those transformed cells to make sure that they don't spit the plasmid out. So we're not going to need a whole lot of culture. We don't need a whole lot of cells, but larger preps like MiniPreps or MaxiPreps will, of course, require larger cultures. So the first thing that we're going to do once we have our culture of cells is that we're going to spin the cells down so that we get a nice pellet. We're going to remove the broth that is on top of it, and then we are going to resuspend those cells in a resuspension buffer, and then we're going to add a lysis buffer to that. So you by now should know what the word lysis means. We're going to blow the cells up, we're going to disrupt their membranes, and all the intracellular contents are going to leak out into the buffer. And that includes the plasmid. So at this point we have a tube that just has the buffer in it, it's got plasmids in it, it's got proteins in it, it's got genomic DNA in there, which we definitely do not want. We just want the plasmid DNA here and not the chromosomal DNA. We've got membranes, so we've got a whole bunch of gunk in here, and what we want to do now is we want to get that plasmid just by itself and not any of the other waste products. So next we are going to add both alcohol and a neutralization buffer. The alcohol, as we've said before, is going to decrease the solubility of the chromosomal DNA, so it's going to become insoluble, and we should be able to then pellet it. And then we'll add a neutralization buffer, because that lysis buffer, in all likelihood, in most methods, contains a lot of sodium hydroxide, so it's very basic. So the neutralization buffer should be fairly acidic so that we can bring the pH back to normal. So all of this is going to cause the plasmid, once we spin all the gunk down, including the membranes and the genomic DNA, the plasmid should be in the supernatant. In this particular method that's shown here on this chart, it's not really shown, but most MiniPrep methods will utilize what are called DNA binding columns. So these binding columns are made up of diatomaceous earth, which actually does a pretty good job of binding to DNA. So we can basically run our supernatant through that binding column, and the plasmid molecules should stick to the column, and everything else should run through. And then from there, we can just break the plasmids off of the column pretty easily so that we get mostly pure plasmid DNA. So here's a picture that actually shows you what one of these kits look like. This one is being sold by Qiagen, which we mentioned before. They usually come in boxes like this, so you'll get some spin columns, so you can actually see these have the DNA binding matrix that we talked about before that's made of diatomaceous earth. You can see some provided buffers here, including the lysis buffer here, the neutralization buffer here. It's a little hard to see what this one is right here, but this is a binding buffer here. This is probably a wash buffer here that contains alcohol. The binding buffer here that will promote the DNA binding to the column. So everything should be, if you will excuse the turn of phrase, everything should be pretty idiot-proof. So this should come with a pretty easy to follow set of instructions. So, and like I said, this is a process that should not take more than 30 minutes of your time. Okay, so let's say that we extract our mini-prepped plasmid DNA. So here's the thing. We think we have DNA. We think it's the plasmid that we want, but how do we know? Do we really know? No, we don't. So there are a couple of things that we need to do at this point. Number one, we need to confirm the identity of the plasmid. We need to make sure that in the MiniPrep, we actually extracted the plasmid that we think we did. So one of the easiest ways to do this is to do what is called a diagnostic digest. So if we digest the plasmid with a particular restriction enzyme like BAMH1 or EchoR1 or HinD3 or PST1, things that we've talked about in the past, then we should be able to confirm the plasmid if we know the sizes of the fragments that would be produced by that enzyme on that particular plasmid. A different plasmid shouldn't produce fragments of the same sizes just because those restriction sites would be located on different places in that plasmid, a different plasmid, and you would therefore get fragment sizes that are a little bit different. The next thing we need to do is we need to determine the concentration and purity of the DNA. And in all honesty, we'd probably want to do this step before number one here. Number two is a pretty simple and quick step. Wouldn't hurt anything to go ahead and do this before we even know if it's the plasmid that we want. So just being honest there, you could probably do step two here first. So to determine the concentration and purity of the DNA, we don't really need to say a whole lot about this. You would just do a lot of the spectrophotometric techniques that we talked about in Chapter 7. You'll get the UV absorption of the sample at 260 nanometers. You can figure out the concentration of the DNA that way, and then you'll also want to get the 260 to 280 ratio to know how much protein contamination there is there. So MiniPrep DNA should have a 260 to 280 ratio in the neighborhood of maybe 1.7, which is fine, but it's not quite as pure as something like MiniPrepped DNA, which should be more in the 1.8 to 2 range. So just a reminder, since DNA absorbs at 260 nanometers, if 50 micrograms per milliliter DNA gives you an OD260 of 1.0, which it usually does, then you can just use some pretty simple cross multiplication to determine the concentration of our MiniPrep plasmid, depending on what absorption you get there. So if you need more information on that, you can go back to Chapter 7 for that. And like I said, the 260 to 280 ratio can be obtained to assess purity. Like I said, MiniPrep DNA may not be quite pure enough for certain applications, but most of them they will be. But if you do need purer plasmid DNA, you may want to consider a MiniPrep. All right, that is going to do it for Chapter 8. I thank you, as always, for your attention, and I look forward to seeing you next time when we get into Chapter 9 and we start talking about the process of actually purifying recombinant protein from cells. So thanks again, and I will see you next time. Bye-bye.